The overall goal of this procedure is to stimulate neuronal cells cultured with gold nanorods using a near-infrared laser diode. This is accomplished by first preparing the gold nanoparticles at the right optical density. The second step is to culture the neuronal cells and add the nanoparticles to them. Next step is the laser irradiation. The final step is to verify cell differentiation through beta-3 tubulin expression. Ultimately, confocal microscopy is used to show the intracellular calcium transients.
We first had the idea for this method when we were looking for a way to stimulate action potential in single neurons amongst a cell population. Demonstrating the procedure will be Jamie Main, an undergraduate student from our laboratory.
To begin this procedure, place a cuvette of gold nanorod solution in the UV visible spectrophotometer. Measure the initial optical density by recording the absorption values from 300 nanometers to 1000 nanometers with a resolution between 0.5 to 2 nanometers. Prepare a 1 milliliter stock solution by diluting the initial gold nanorod sample to an optical density of 1. Centrifuge 1 milliliter of the gold nanorod solution twice to remove any chemical excess. Then, remove the supernatants and resuspend the gold nanorods in deionized water. To prepare for use in cell culture, sonicate the gold nanorod solution for 5 minutes and then sterilize it with UV light for 30 minutes.